Okay, this is the last last set of slides for this unit session on chemical basis of life. So we're just going to now talk about various nutrients that we have in our body. So carbohydrates is the first one to talk about. So it's often short form CHO. It literally means hydrated carbon. And again, we're just going over the general facts about it. It contains carbon, hydrogen, and oxygen, with the hydrogen and the oxygen in a 2-to-1 ratio. So 2 hydrogen to 1 oxygen. I've just written glucose down there. Now you're never going to have to write these chemical structures or draw them or anything like that, or these I just want you to understand. Some people like to know what the differences are in terms of how they look if they're written this way. So they're classified according to their size and their solubility. In other words, a monosaccharide would be the smallest size. Disaccharide would be double. Okay, and our basic structural unit here is a monosaccharide. The larger the molecule, the less soluble it will be. And again, just to show you this structure, but I'm not ever going to have to ask you to draw it, so don't worry about that. And again, just a monosaccharide, a disaccharide is 2, and a polysaccharide is many, for sure. Sugars. Lipids have 2 types of building blocks. I'm going to go to the next slide, okay, and they have a fatty acid, okay, chains, you see that here, right, and they have a glycerol portion here, okay, and they also are composed of carbon, hydrogen, and oxygen, but the oxygen is in smaller portions. And generally speaking, lipids have 3 fatty acid chains with them. Now we'll talk about some differences there shortly when we talk about different types of fats. And we will talk about this in a couple of slides, but this is generally a lipid molecule. This is the phospholipid molecule, and you see that we add a phosphate portion to it. Proteins also contain carbon, hydrogen, oxygen, and nitrogen. And the amino acids are structural units. There's 20 types of amino acids in our body, and all of them contain 2 parts, 2 structural parts. I'm going to go to the picture. Okay, so they have a nitrogen group, which you see over here. Okay, a nitrogen group, and they have a carboxyl group, or COO, and then above it is some sort of other molecule that makes it specific. So you can see here's the nitrogen group, okay, and here's the carboxyl group, okay, and then this is what makes this cysteine. Okay, you're not going to have to memorize all of those. All I want you to do is understand structurally how they're created. Nucleic acids are the most fundamental components of the cell, DNA and RNA, and they have a huge role in our cellular activities. This is where our genes are stored in our DNA, so it's our blueprint. Also contain carbon, oxygen, hydrogen, and nitrogen. I'm going to go to the picture too so we can look at it. So they have a building blocks, or what we call nucleotides, and this is where you can see one here. Okay, so we have a base, okay, some kind of base structure. This is the sugar right here, which is either going to be ribose or deoxyribose, depending on what we need. And then they're all connected with a phosphate bond. Okay. So showing you this picture here, okay, we'll see, you can see that they share this bond kind of like a ladder all the way down, and then these bases are connected in the middle. Okay. And we're just looking at that generally. We'll talk more about DNA and RNA in the chemical basis of life, or pardon me, the cellular metabolism model. Okay, so fats. So there are and when we look at a triglyceride, its building blocks are glycerol and 3 fatty acid chains, which we looked at previously. Other fats have a different combination of the fatty acids, and phospholipids and steroids are also examples of fats. Saturated fats, and we're talking about triglycerides now, are when the carbon bonds are saturated. In other words, they're very structurally sound, or they're happy. Their valency is such that they're not going to separate. And generally speaking, in our diet, these are foods that are solid at room temperature. Not always, but most often, like animal fats. So the marbling in meat, butter is a good example. Okay. Unsaturated fats then have some double bonds, so they can rebond or they can separate and not be so solid inside of us, if you will. And they're often liquid at room temperature. So peanut oil, corn oil are good examples of that. Phospholipids, second kind of fat, have a similar structure to triglycerides, but in replace of one of the fatty acid chains, there's a phosphate group. And these are very important. Your cell wall is a double layer of phospholipids, and so it creates important cellular structure for our body. Steroids have, once their structure is such that they have 4 rings of carbon atoms, and they are examples include vitamin D, bile, cholesterol, and various hormones inside of our body that we use. So cholesterol is ingested with animal products. Okay, we make cholesterol inside of us as well, but the cholesterol that we eat, okay, comes with animal products. So we think about, steak and butter and that sort of thing. So because they are saturated, and they can contribute to the developing of what we call arteriosclerosis. So sclerosis means hardening. Arterial refers to arteries. We get about 15% of our cholesterol from our diet, and 85 is converted in the liver for our own needs. So we make some and we eat some. So sometimes when people go on a low cholesterol diet to try to decrease their cholesterol, they don't always achieve that goal because, of course, their own body is making it for them. How do we measure this? We measure it through HDLs and LDLs, high-density lipoproteins, which are good because they go around and collect cholesterol for breakdown and take it to the liver. So I always call them happy fats, the happy lipids, lipid proteins. LDL, low-density lipoproteins, because they tend to stick to the artery wall and they don't go to the liver and get reorganized. There's a couple of articles posted on your webpage about that. Differentiate between essential and non-essential amino acids. Okay, so proteins are digested and they're broken down to form amino acids. Okay, so if we eat a steak or beans or whatever we're eating that's protein, when they are being catabolized, I guess, they would become their basic structure. Our circulatory system absorbs them. Okay, the first place they go is to the liver. I'll show you a picture in a minute about that, and then we send them out to the general circulation later. So this circulating amino acids, once they're circulating, they're quickly picked up by the body cells, and the body cell tries to keep a stock of them, sort of like a pantry, if you will, and they try to always keep all 20 of them in your body, and whenever we need to make proteins, we need to be able to rely on having them. So of these amino acids, 8 of them are termed essential. It means that we can't produce them in our body, or we can't synthesize or build them in our body. We must ingest them. Just to help you show you what happens when we eat our food. These villi, this is just one, that are in the lining of the small intestine. And you can see that up the middle here, is what we call a lymphatic capillary. That's going to absorb your fat. But surrounding it here like this is a capillary. And that capillary absorbs through the cell wall, absorbs the nutrients and takes it in. In this situation, they go into the venous side. And the venous side then takes it to the liver before it goes back to the heart to be reorganized. The next picture is going to show you that better. So when our stomach and our small intestine, what happens is they're going to go to the liver first, amino acids, and then the liver is going to say, okay, I need to keep so many of these. And then once the liver has what it needs, it sends it on to the heart. And then from the heart, it sends them to the body so that the cells can pick them up. Okay? So we've talked about essential, okay? The ones that we can be made or we can synthesize in our body are called non-essential, and we can either get them through our diet or we can produce them. One source for essential, if we're looking at the essential ones, the 8 that we talked about earlier, they can only come through diet. All proteins are made up of a combination of both of essential and non-essential amino acids. And what's important is that whenever we're going to build protein or protein synthesis, we need to have a pool of both types ready. We make a number of proteins in our body, and they're structural, which is building blocks, or functional. And functionals are they're the proteins that have jobs, like hormones, like neurotransmitters, things like that. The protein is rated to the content it has. So all of the food that has the 8 essentials are called complete proteins, and they come from animal sources. We have to get them that way. Foods with only limited or inadequate amino acids are called incomplete and most often plant sources, which is why vegetarians have to balance how they get. So if you ate an animal source protein, you're going to get all of the 8 essential. If you're a vegetarian and you're careful about how you manage that, then you have to eat a combination of certain foods to produce the amount of amino acids to get those 8 essential in there. Again, these are the ones that are not made by us. So that takes us to what we call nitrogen balance. Nitrogen balance is looking at how we build proteins versus breaking them down. So it's how we incorporate versus our loss. So obviously we always want to have balance in the body of protein. So if the body is in balance, then the amount of protein we're taking in and incorporating into our cells is equal to that of what we're losing through our exit points. If one is in a positive nitrogen balance, then we're saying that protein synthesis or building proteins is greater than the rate of breakdown and loss. So we think about growing children, we think about pregnancy where a new individual is developing. We're hoping that after trauma or surgery, the tissues are being rebuilt, which is why we always want people to be on high protein diets in those cases. A positive balance means that we've got more protein being incorporated into the tissues than what is being broken down or lost. Meanwhile, negative balance is protein breakdown, and it exceeds what we're using for building. Okay, remember structural or functional proteins. Structural are building blocks like cell walls, things like that. Functional are the proteins that have jobs, antibodies, hormones inside our body. So how can we have a negative balance? Could be emotional stress, physical stress, poor diet intake, or for some reason our tissues are losing more than they're being replaced. So that's a negative balance. Negative. So that's the end of the clips for this session.